we need to understand is the mammalian bone structure. There are several types of structures in bones. Woven bone is laid down quickly, approximately 4 micrometers per day, and is present in fetal bone during fracture repair and in some pathological conditions, such as neoplasms and infections. Woven bone consists of irregular organization of collagen fibrils. Lamellar bone is laid down slowly, 1 micrometer per day. It can be primary, which is laid down de novo, or secondary, which replaces the existing bone. Lamellar bone has organized structure that consists of thin layers called lamellae, which are successively laid down and are approximately 3 to 7 micrometers thick. Fibrolamellar bone is simply a combination of woven and lamellar bone. It has alternating sheets of lamellar and woven bone. It is laid down more quickly than lamellar bone and is found in mammals with bones that grow quickly in diameter. Plexiform bone is formed more rapidly than lamellar bone. It appears as the layering of rectangular primary vascular canals in a well-organized network known as plexus. These rectangular sections are layered in a horizontal direction. Plexiform bone is typically cited as a form of bone structure that is indicative of non-human origin, but it is not prevalent in all non-human groups and can occasionally occur in rapidly growing human infants and children. But mainly it is present in large, rapidly growing animals such as cows, horses, and sheep. In humans, fibrolamellar bone is laid down first and is then replaced with primary and secondary lamellar bone through process of modeling and remodeling. Remodeling, especially with the bone cortex, involves formation of Haversian system with secondary osteons. In Haversian system, concentric lamellae are laid down around the vascular Haversian canal in the center, connected by transverse Volkmann canals. In between osteons are parallel lamellae, interstitial bone with star-shaped osteocyte lacunae, which are basically captured osteocyte cells, connected with canaliculi.
The human skeleton gives the body its shape and provides physical support for the systems contained within. It also forms part of the musculoskeletal system that enables us to move. The structure of bone is optimized so that it is strong but relatively lightweight. The interior of bone is composed of bone marrow. It is surrounded by two major types of bone tissue: cortical bone, or the hard outer shell of bone, and trabecular bone, the spongy-looking center. The amount of each type of tissue in bone is dependent on the function of that bone. The basic unit of cortical or compact bone is the osteon. It is composed of successive concentric lamellae. This structure contributes to bone strength by resisting bending. Cells called osteocytes are distributed within the concentric lamellae. Osteocytes form a complex network that is thought to be important in maintaining the viability and structural integrity of bone. At the center of the osteon is the Haversian canal. These canals contain blood vessels and nerves. The blood vessels within bone facilitate the exchange between osteocytes and the blood. Trabecular bone is present in the interior of some bones and resists compression. Osteocytes are also contained within its structure and again play an important role in sensing local changes in strain. Trabeculae are covered in a layer of flattened lining cells that are thought to be involved in the dynamic process by which bone is formed and broken down. Bone marrow is found within the interior of bones. The surrounding trabeculae and vascular network provide structural support, nutrition, and a waste removal system for the heterogeneous group of cells found within this space. Bone marrow is a site for hematopoiesis, the process by which the cellular components of blood are formed.